this is her STEM project. It was her great-grandfather, great-great-grandfather, and other people. It's not mythical bones of my ancestors. It's my great-great-grandfather. My mother's father's father is buried there. And so the state wanted to come in and put a toilets and all the shit on the bones of my ancestors. So the people obviously got very upset. They had already been upset that they had been allowing people to park here on the graveyard. And so one day, one holiday, I forget what it was, 4th of July, they were having a party out there, the local Hawaiian people, and they got upset because all of these trucks were coming and parking in the graveyard on the bones of their ancestors. So they basically just sat in the middle of the road and had their luau in the middle of the one and only access road right here, and blocked traffic. And so the police were called and everyone were called, you can't do that. And they just said, we've had it. You're not gonna park in our graveyard anymore. And they threatened everything, and the state backed down and put up fences and prohibited parking. So the state recognized this was a sacred spot, and that the Hawaiian people, they claim they still own it. Okay. So now, move ahead. We got it all worked out. They're gonna give us some benefit of being sacred, and what's coming up now is you're gonna build a great big restroom facility right on our graveyard. Not a good idea. So they got very imaginative, and what they did is they put the new restroom facility right on top of the old one. They brought in the kapuna and asked, where can we do the American Disabilities Act sidewalks? We don't want to put a sidewalk on top of anybody, and we're not going to go deep, but the sidewalk only goes that deep, but just tell us, where can we do it? So we got a sidewalk here. And then, what are you going to do with all of the waste products? What we're going to do, we're not going to put dump raw sewage on Iwi Kapuna. We're going to clean it up. So we're going to put in a septic tank right there. Okay, septic tank. And then out of the septic tank right here, we're going to build a constructed wetland. It's about the size of this room. And they're going to rubber line it. And they're going to plant native Hawaiian plants in it. And the nutrient-rich water coming out of the septic system will go into this pond, and the plants will take out all the nutrients, nitrates, phosphates, and then they have a leach field that goes out into here, and all the clean water will go on the bones of your ancestors. Not dirty water, clean water. And isn't that and we promise we'll plant native Hawaiian plants, okay? So they built it there, but the local people still noticed that there was he sort of smelled something down here, and there was a lot of algae growing really healthily right in here. And when it rained a lot, that it flooded that whole area, so the whole leach field was underwater, and that it all ran just down the road, right here past the lifeguard station into the beach. And it was really pretty foul. So when Leah decided she wanted to do a student project, and she was from that area and she was upset about what was going on, I said, here, let's just see how effective that new system is, and whether there's any contamination out there at all. And so she went out 5 times, and we have site number 1. So the water comes out of that pond and goes into pipes underground, and then goes into the leach field underground. So we wanted to get the water coming out. So how are we going to do that? And so I went out there one day, and I found that they have little pipes sticking up that you open up so you can push the junk out in case it gets stuck. And so, admittedly, without any permission, because we never get permission, without any permission or anything, we simply went down there early in the morning before the janitor came to clean anything. We went real early in the morning and undid the top, put a pole way down into the water, got our water sample, and brought it up, put it up, and ran back out of the way so nobody would see us. So we sampled right coming out of that pond, the water coming out of that pond directly. Okay, and then we would sample down here in front of the lifeguard station, sandy spot, and we would sample over here, number 3, which was on a rocky karst, limestone karst, not limestone, but karst topography, hardened sand. And here, that's the sites 1, 2, and 3. And what you can see is that the leach field had high ammonia, it had high nitrates, it had high phosphates, it had way high bacteria counts, and that you did, at site number 2, you did get some high bacteria counts, but there wasn't really very much going on there. And the explanation for that, we think, is this. It's a high energy wave zone. And so the waves are coming in and just flushing it all out. So that's good news. The good news is it's flushing it all out, perhaps. But we still smelt it, you know. So this just gives you an idea. If you're looking at bacteria counts, that was our average count. It was not polluted. Look at the streams are bad. It was not polluted. But if you look at the leach field levels, the ammonia, and so we question, is that constructed wetland thing, is it working properly? And what we found out was, yes, it was, because the Department of Health said all they had to do is measure biological oxygen demand and turbidity. They didn't have to measure bacteria, how much bacteria are coming out. They didn't have to measure anything coming out. And they only had to do it once a year. So they could, nobody was checking on whether that constructed wetland was working or not. And it was all dumping into the groundwater, so it didn't matter. It was right here was when they put the brand new septic system in. And so the mean values during the summer of the number of bacteria went down, but this year it went way up. So I wonder why.
El Nino year?
Huh?
El Nino year, maybe? With all those rains that we had, the ponds just flowed over, and now all the nutrients that were supposed to be eaten by the plants to grow flew out with it, maybe?
What's the source? What's the source of the bacteria that we're measuring? What do we think the source of the bacteria is? What? Human poop.
Yeah.
Did we have more human poop that summer? Was this a banner year for tourism on Kauai? Now, I don't know if I have this data. I'm not sure. How do you, okay. So, I talked to this about, how do you measure whether it was a banner year or not? You measure how much water that park used, right? And so way back, they gave me this. I emailed the guy last week and asked him if I could have the most recent data, and he has not responded. So I don't know. But you can see that's how I'll find out whether there was a lot more tourists out here. The other thing that's happened is those beautiful native Hawaiian plants that they put in that constructed wetland, they all died. So they're not, we're looking at what is in that leach field. They're not sucking out the nitrogen, and they're not killing off the bacteria. That constructed wetland is a scam. It's not working. And because the plants aren't growing. But the other thing is that whole constructed wetland was designed for 2,100 gallons per day. Okay, here's 2,000. And these are what? Monthly averages? Right, monthly averages. So it's way above capacity every single day, except this month, which probably meant it would probably rain like mad, and not very many tourists went out there. Okay. So the system, because of so many tourists, they're getting twice of what the system could handle according to the design specs. But the thing isn't working because all the plants are dying. So we have a real disaster going on out there with tremendous amount of nutrients being dumped on Iwikupuna and also hurting the environment that way. While we're in the midst of doing all this, two really important things have happened. One is that that area at KA has been designated by the state as a community-based fisheries management area. The state has abrogating power to the local community to set certain areas for no fishing and seasons, and also closed entry. You can only fish there if you have our permission. They're hoping that by doing that, the whole population of fishes are going to increase. What they're not counting on is that we're getting a tremendous amount of pollution coming in from that, right into that area, which is causing the algae to grow. I haven't gotten there yet. Okay, causing the algae to grow, which we'll find by the nitrogen acetate, but it's also putting other chemicals in the water. The other thing is that we've been working with the community group to make that park a community-run park. The community who has lived there for 1,200 years have never given up title to that. That's their property, that's their graveyard. The state claims they own it, but that's where my grandfather's buried. And so we've been working with them, and they're going to take over management of it, and they're going to control and limit the number of people. And so I've been brought in and dealing with this, and one of the first things we're going to do is we're going to move the restroom, build a new restroom facility so it's not all on the beach. And we're going to build it much greater capacity, and we're basically going to ask everybody to go to the toilet before they go to the beach, so that it's way back inland, so that it should not have as great an impact. So I'm really interested to see what the water usage here is going to be. How many people, how many flushes a day? It's sort of weird when you get down, your science gets down to flushes per day. Common denominator. Okay, the next slide. So you saw what we did for Hanalei. We did the same thing. This is the analyses were done here, but downstairs, you were saying?
Just right
next door. Right. Yeah. I've never been there. I sent it all off here to be analyzed, and the big thing is right here. If you go perpendicular from the leach field, the shortest distance to the beach at site number 3, and the bacteria or the nitrogen isotope concentrations are right there at sewage level, according to the people I need. And the algae is dark green, it's beautiful, growing, and so I have no doubt that the bacteria may not be getting out here, but the fluids are getting out into that lagoon area, and that in that one spot where it comes out. Now, I'm a field biologist. I'm not an analytical chemist or anything like that. And I like going out in the field. The reason I'm doing this science, the reason I became a scientist, I like being out in the ocean, I like being out in the field. And the amazing thing is, the more you go out in the field, the more you see. And so one day I happened to go out there at a really low tide. And what did you see? You saw all the water pouring out. It wasn't you don't need to be a PhD to say, hey, water's pouring right out into where's it coming from? Where's the closest place? The closest place is the septic tanks or the leach field right there. And that's right where that algae was growing. I didn't bother, and for this student project, there's no need for us to go in and measure the flux rate or anything else. We've proven our point that there is a direct linkage here between the heavy use by the park and the restroom facilities right there. And so my recommendation for this new park is move it as far away from the ocean as you can and design a better system for handling that. But the septic system problem, we're going to get into this. Septic system problem is septic systems only take care of basically the bacteria. They don't take care of the nitrogen, they don't take care of the phosphorus, and they don't take care of the acetaminophen, DEET, trichlopor, all the other chemicals that come off your bodies. What's next?
Do you know what species
Sure.
of algae it is?
No.
Okay.
That's an easy one. So I gave it off to somebody to analyze, but I don't know what it is. The interesting thing in the knee paper, and also the one by Dahlia et al. That's there, is that it doesn't matter. It doesn't, when you have that big a difference, it doesn't really matter.
True. Yeah, when is the bathroom expected to move? Do you know when you're? Think you might choose? I'm interested.
No, the reason I don't know is we just presented a master plan. We did an EIS and presented a master plan to the community, and all of the young people in their 20s, 30s, who've never been part of this thing, that never listened to their kapuna, they all got all up in arms about, what are you gonna do, put another bathroom? And so we probably have another 2 years before we're gonna be able to do it. Meanwhile, we're getting food. So this would be a great study, you know. Just like before, we were able to get some samples beforehand and after. Just, yeah, does the thing work? No, the thing doesn't. It doesn't. So this is an example of human contamination via septic systems and stuff like that. But what I'm going to move into now is agriculture. That was my title, right? I was supposed to look at groundwater agriculture and urban development. Okay. So all of these colors, this is Waipa in Hanalei Bay, and all of these are fields of taro, and this is the ocean, and here are sites 1 and 2, 1, 2, 3. The water's coming from Mauka, coming down in. Have all these crops. The water comes down here through this canal, and then it goes down here. It goes out this canal here, site 8, and then I put a well in, a 2 meter deep groundwater well right here, right off the ocean, right next to pond number 1. So what you're going to see is mostly ditch water and then the well water. Now, the question I had was, from my nitrogen isotope work before, that we're getting a lot of fertilizer into the ocean, causing a lot of algal growth right there. And there's a reef right here. So how can we cut back on that getting into the ocean? So my first thing was to convince the farmers that they shouldn't throw their dry fertilizer into the wet pond fields and let it dissolve and percolate, but rather that they should till it into the dry soil so it was incorporated in there, so it didn't get flushed out when they turned the water on. The other thing I went into was, let's use slow release fertilizer in the soil, and so they can maximize the uptake of that. And that's what all these different colors are. And so the idea behind this was that all of these fields, they dig them out and they pack them all down, but the water percolates through the pond. They're not lined. It percolates down and goes into the groundwater. That's my hypothesis. It also I know it goes all to ditches because the pipe comes out of the pond into the ditch and they dump it all out. And they have to have water flowing through in order to grow taro.
Hey, Carl, where is this? Where is it?
This is Hanalei Bay, the west side of Hanalei Bay in Waipa.
Excuse me?
Ahupua, Waipa.
For the slow dissolving fertilizer, don't you need other chemicals to keep the fertilizer from dissolving fast?
What they do, what I'm using is something Osmocote, or it's like orchid fertilizer. It's a plastic, some polymer bead, and it's all filled with the whatever you want at whatever concentration you want. So I got nitrate, I got phosphate, I can get combinations of those. I can get a 30-day, 60-day, 90-day, 100-day.
Right, I agree.
Yeah. And so the question that I wanted to ask was, by doing these best management practices, could we reduce the amount of fertilizer going into the ocean and affecting the marine environment? And the way I did it was to get this taro farmer to try the different kinds of slow release fertilizers and stuff. And his concern was, you're asking me to try these fancy things. What happened if my crop fails, right? If my crop fails I'm gonna be out thousands of dollars, right? And so, I went to him, and I said, trust me. And he goes, right, I'm a businessman. What happens if my crop fails? And this comes at a point in doing science where you have to put up or shut up. And I said, I will guarantee you any monetary loss, because I am sure from my calculations that if you use this stuff, you're gonna get as much crop, if not more, and we're gonna clean up the environment. And I should add, the farmer here is a active waterman. He's Howley, he grew up there, and he does my water sampling for Surfrider right out there on that reef. He's really concerned about the environment for himself and his kids, who now kids help do water sampling. So, the question was, could we do it, and could we still do it profitably? So here, this is the water coming out in the ditches over time, and what you're looking at is the ammonia concentrations. And you look, so I started it off, and when I first started, they had regular fertilizer use, and then they switched to the other kind. And guess what? It all went up. The amount of ammonia went up. But what I showed you, what I didn't count into fact, was that the water was coming in the ditches, was coming from Malka, from the other guy's taro fields, his brother's taro fields. And it was coming from the river, from pipes into the fields. And when you do your ammonia test, you have distilled water, and you add your chemicals and colorimetry. There's something in there. The chemicals gives you a reading. So here, what I did was I subtracted out the water coming in the pipes, the water in my test tube, and the water coming through the ditches, Malka. And this should be just the influence of the taro fields, not the water coming into the taro fields. And if you look at it that way, you have over time, there's a decrease in the amount of ammonia in that ditch water for both of those downstream effects. Now there's also a cyclicity in here, because he is fertilizing the fields in different times. But this is a nice correlation, lower trend, yeah.
Do you consider the conversion of ammonium to more oxidized forms? So do you look at total nitrogen also?
The reason I used that was I could get a cheap hot colorimeter to use in the field, which was way better than any kind of tablet and color thing, but not laboratory grade. So it was, and the grant was only $20,000. This is the well. And so the upper one is the well water, not counting the surface water input. And there's not much change here. The bottom one is taking out the surface water input. And what I'm finding, we have the next one. So what you see is that the amount of ammonia is pretty much, net ammonia is the same over all period of time. And here's what I found is that if I look at before I went to slow release and after I went to slow release, okay, for the ditches here, there was a remarkable change in the amount in the ditch water coming out. There was not much change at all in the well. And here are the p-values, they're not significant. This is marginally significant here at a 10% level. So the question was, how much stuff was actually coming in from groundwater as opposed to stream water? And this slide, I just put it in today. Berg helped me because, I should have put that one in. This shows you over time, it rains a lot. And the idea is if it rains a lot, that one, the taro fields will overflow the water will pour out and go down the ditches, but also if it rains a lot, it fills the pond and it's gonna push that water down into the groundwater. And it's raining back there, the whole groundwater is gonna increase. So one of the things, and I haven't looked at this data probably in 2 years. Look at it, this was 4.5 inches of rain in the past 24 hours. I was waiting for rainfall events. So this was the study, the blue ones were the study before I started my study. Then this were the boxes and the triangles were different years of that study. The important thing here is that there is a decrease here. There's no real strong correlation. But here, look at these counts. What happened here? There was no rainfall. What happened? And this is well water. So I got this, I'm used to getting counts at 0.5. We're doing it right in the field, right? We take the water out and do the test right then, fresh. This one's coming up at 2.25. And we had to dilute it out. We had to do it about 4 times, dilute it out, because we didn't measure that high. What the hell's going on? And so I asked the taro farmer, I said, what's going on? What have you done different? Did you just kill a horse or something? He did kill a horse before, and that changed my bacteria counts. But what's going on here? He said, I didn't do anything. And I was under strict control. He was going to apply the fertilizers that I bought for him in a strict fashion, right? So he said, honestly, no, we didn't do anything. How did that happen? And he said, why don't you talk to my brother? They didn't talk to each other. Why don't you talk to my brother? His brother had 5 acres of taro Malka. And his brother was cantankerous. So I went back and spoke to David. I said, hey, David, what have you been doing? I said, did you just fertilize? And he goes, yeah, I just fertilized. I said, how'd you fertilize? He said, well, I had a huge amount. I got 10 bags of thing and I dumped it all in that pond and that. He super over-fertilized in the old-fashioned way, flooding the pond and then pouring in these pond fields. 4 days later, I get this spike. So the water is moving really fast, about a half a mile, from back there, downslope, groundwater, underneath all those ponds, and then my well is only 100 feet from the ocean. So I said, this is really the dynamics here. Now, this is where you turn it over to somebody who does groundwater discharge. I don't do groundwater discharge. I dabble in other things. This is fascinating. But what it shows to me is how great an impact agriculture, taro farming in this case, sacrosanct, culturally important taro farming, done by all the Japanese, are affecting the groundwater in this area. And it's what we found from all of this is that the slow release fertilizer, I would get more pounds per acre using slow release fertilizer. Okay. I could use less fertilizer, therefore I could use less fertilizer and get the same yield. Follow me? Less fertilizer, use the same yield. The less fertilizer you put in, the less pollution you put in, the less you're going to get out. Whatever you try to do to clean up the pollution levels are pretty ineffectual. I put floating trays of plants in there and was able to get out a huge amount. But I think I measured, I was getting 2 pounds of nitrogen a day out of that stream from all the fertilizer use. What I was measuring, 2 pounds of nitrogen a day dumping into the ocean. So you can't really, it's almost impossible to clean up pollution. Talk about CO2 and global warming. It's almost impossible. So you don't do it in the first place. In this case, you can do it by simply putting 25% less of the fertilizer into the ponds in the very beginning, and it'll clean it up. The bad news is, bad news is that it costs about 40%. Well, I don't know what it does now because oil has crashed and the fertilizer is all made out of oil, by the way. And so the price probably has crashed. The other thing that has happened is that Asia is using slow release fertilizer. All the rice fields, the massive rice fields, they saw they were losing money dumping all that fertilizer on. So people are inventing slow release fertilizers, and they're going to massive production scale, which is going to bring the price down. That's good. So I haven't followed it in the past 3 years to see what the price is. But when I got done with this, the answer was it was not cost effective. The other problem was the slow release fertilizer is encapsulated in little plastic beads. Have you heard about microbeads recently? Yeah. These are mega beads, they're not microbeads. Microbeads are what's in your facial products and toothpaste that are being found in the Great Lakes and oceans everywhere, polluting them with plastics and filling up the guts of fishes and corals, by the way. And these are bigger. And so when it's all done, they don't break down right away. And so my friendly taro farmer, I showed him that he could use a lot less fertilizer. He could pollute the ocean a lot less. And he said, yeah, but Carl, I'm a surf rider. I ban plastic bags. We're gonna ban styrofoam, and you're telling me I should put plastic beads in my fields? So it hasn't gone anywhere. I haven't pushed it any further. It's very effective, but not with the technology we have now.
Carl, do
Yeah.
you know that your hack colorimeter test, does it convert all forms of nitrogen to ammonium? No. And then you test for that? Or
No, it doesn't.
It just tests straight up for ammonium. Do you know what the pH was?
I don't know. I don't know. Do I? No,
The closer
I don't for these.
Okay, the closer you get to 9, at 9, all ammonium is in the form of ammonia. Fish start floating upside down.
Yeah. I was trying to think. Back here, I don't know. Back there we had it. The pH level. The pH levels there are. I have all that data in the paper because I measured it all in the soil. Because I was looking at how the soil was regenerated or depleted by the thing, but it's always about 8. I think.
I know it's rough in the field to pull that off, and on Kauai you just don't have the lab resource. But for most of us, I mean, we know that those fertilizers have percentages of nitrate, urea, which is then converted into ammonium and ammonium nitrates.
In this case, I was not as concerned as the absolute value, and it's the same with our pesticide, glyphosate levels and stuff. The absolute value is not as important as the comparative relative values between treatments, whether it has a treatment effect or not.
But different kinds of fertilizers are going to be formed of different constituents of ammonium and nitrate.
Right. And what I used here was what the farmers were using. So there was 3 different colors. It was different kinds.
Okay.
Now, switching. So what I've been talking about so far really has been nitrate and phosphates and then bacteria and human pollution. But there's lots of other stuff out there. And the question comes up as an environmentalist looking at pollution, what should you be looking for? And if you are worried about an oil company dumping oil, you go look for oil. If you're worried about sewage, you can look at sewage. You can look at the bacteria. You can look at a lot of different things. And so I had a couple of high school students who wanted to do a water quality project, and we were trying to think of what they could do, something new and different. And the question came up was, how bad are all of the streams on Kauai? How polluted are they? And the question, how do you measure that? So they went out on a couple of weekends, and they sampled from the west side, number 1, west side, all around the island up to the North Shore. And there were 19 rivers and streams that are easily accessible by the ocean. And they sampled each and every one of them. And then we did this series of tests with very simple test kits, high school level test kits. We measured nitrate, phosphate. I then discovered that Hawk made a kit for determining detergents. Detergents are a class of ionic compounds manufactured by just a couple companies, and I repeatedly asked, what are they testing for? What? And they wouldn't tell me. It's proprietary. You're just testing for detergents. Okay. I said, fine. And then we tested for the bacteria. So we tested for these things, all of which should be indicators of wastewater coming from a house. So nitrates and phosphates are coming from the fecal matter and the urine. Enterococcus bacteria is coming from fecal matter. And then detergents is from washing your dishes or washing your clothes, and shouldn't be anywhere else. I then tried to figure a way to put them all on one graph, and we invented the term Student Household Wastewater Index. And how we got that is we measured the nitrate and the phosphate. The detergent level, we multiplied it by 10 to get it up into the same range. And the enterococcus level, we divided by 100 to get it in the same range. And then stacked them all up. And basically all of our streams here are polluted with the enterococcus, except here. They all have phosphate, and where's the phosphate coming from? Well, our soils has phosphate in it, so it doesn't necessarily mean it's sewage. Nitrate levels, most of that sucked up, and we only found one here that had a high level. But the detergent one was really interesting because that should only come from households. And if you simply stack them up in this, the index would suggest that those that have the higher levels would be much more polluted by human waste than the lower levels. And in this case, we're picking 4 different indicators of household water pollution. I think it's really cool. The girls did really well at the science fair with this until they got to the state level, and the guy said, well, how many replicates do you have, and what are the statistical differences between all that? And they said, that wasn't the point. The point is we invented this index for water sampling to allow the government to go out and test this. So it didn't go too far, but we had a lot of fun in doing it, and I think it's really indicative of, if the question is, are our streams polluted, what do you look at? And you can look at the nitrogen isotope, you can, detergents, that's a pretty neat thing to do. There's optical brighteners, right? Do you know about that? Optical brighteners that are in detergents also, and there's a whole easy way of testing for optical brighteners in water too, which only would come from laundry detergent.
What's your cost per sample on that?
Pardon me?
What's your cost per sample for that method?
Cost per sample?
Yeah, to run those 4 tests on one sample, how much were they?
I don't know.
I know hack kits run usually about $10 a test and tube is the terminology they use.
Yeah, but this one's more. You have
For
to
the
use
detergent.
chloroform and all sorts of stuff. But it was only like $25 maybe. I don't really remember, and Surfrider footed the bill for it. The enterococcus, we're using our supplies, cost us about $10. Bacteria, and then the nitrate phosphate are only like $1. So let's just say 15 plus the 25, the $40 a shot or something. Probably less. So, sewage water contains lots of stuff other than nutrients and bacteria. The state of Hawaii, as part of a broader U.S. Geological Survey of streams and waters nationwide, went out about 5 years ago and got samples for the USGS and sent them off to them, and they did analysis for all sorts of chemicals. The Department of Health, got the results a year later or something like that, and invited me over to discuss these results, okay? And they didn't send them to me because they're hush-hush. And come on over and let's look at them and tell us, what does it mean? And this is fun. So I went over, and I was having other meetings. I went over, and the guys had printed out the spreadsheets for these, and they had, I don't know what, 15 different sites or something like that, 15 sites. And they opened up the spreadsheet going that way for all the different chemicals alphabetically. And that spreadsheet was longer than that table. It was about 20 feet long. And they said, what do you think? And I was like, what? What? I said, give it to me electronically. And I'll come back in a week. And so first thing was we found most of the stuff we didn't find. So I just, and then I asked them, what are these things? Because most of them were the chemical formula. What the hell is it? And so I spent a whole bunch of time with Google trying to figure out what all these things are. And then where would they have come from? One of them, which I didn't put up here, comes from cranberries. I don't know where, we figured, I don't know where we figured out. Papaya or something. It was weird. Some things we have no idea where it could have come from. But in giving this talk about water quality and stuff, I went back to them and said, okay, you want to keep all this data secret because you don't want people to know your beaches are polluted. But it's really important. So how about, will you let me pick out a few chemicals and the beaches and just talk about presence or absence? I'm not going to tell you concentration. I don't know the concentrations anymore. I'm not going to tell you the concentrations, just the fact that these showed up. So for a lot of the stuff, that's all the important thing is. If you have a measurable amount there, if you have it, then you got it. That means you got that kind of pollution. But what I think is fun is what I picked out on top. Yeah, go ahead.
So for the odd ones like cranberries or papaya, were the levels at a significant amount? Like it's okay they were measurable, but were they significant to actually warrant, like if they had announced and come forward with the data, would it have even mattered? Like
No, because most cases we have no idea what the environmental impact level would be.
yeah.
But they're tracers. That's the important thing. Just like fecal indicating bacteria are, Enterococcus is an indicator. You don't really care what the concentrations are, it's an indicator. These things are another kind of tracers like nitrogen isotopes. So it's not whether the level itself is toxic. And in most cases, you're not going to have those toxic levels in the stream, and you're definitely not going to have it in the ocean because you have the whole damn Pacific Ocean to dilute it out. The only reason our beaches and everything are clean is because we have the whole Pacific Ocean washing waves up on it.
Well, some of these compounds are also subjected to oxidative processes as well as UV. So you might find them in the freshwater onshore, but as they go out, even if they just stay on the surface, sunlight and
Sure.
interaction with the atmosphere can break some of these compounds.
Yeah. Now, okay, so atrazine, which I misspelled, is a pesticide. D, insecticide you put on your skin. Caffeine, what's the number? 16% of caffeine that you pee out 16% of the caffeine you drink.
3.
Something.
The paper said 3.
3%?
According to the D paper.
Okay, 3%.
I just learned that today.
Yeah, but we drink so much coffee, it doesn't matter. Cholesterol, this is soluble cholesterol. Triclosan, antibiotic soap. It's also in your toothpaste and everything, antibiotic. And acetaminophen, what's that, Tylenol?
Yeah.
Right?
So what's the demographics of Poipu and Salt Pond Beach?
Oh, we gotta look at that.
Where do you know the demographics?
Okay, so-
before doing it?
What do you think is important? Okay, so Hanoi Pavilion. Okay, Hanoi Pavilion sample. Hanoi Bay, remember it? It's in the middle of the bay, there's a county beach park. And I don't know, 600 people, 1,000 people a day pee in that restroom facility, and Dr. Berg got the money, and we took out those nasty old cesspools and put in state-of-the-art septic systems. And what you find is that you have caffeine and cholesterol. You also find you have really high counts of Bacteroides RNA out there. It's nothing live, but so those septic systems at Hanalei Pavilion, because of the Bacteroides tests and this, must be pushing that groundwater. Okay, where do they get this? They walk out to about their calf depth. They walk a foot into the ocean with a bucket, scoop up the water, and pour it in the sample jar. So it's not out, it's not groundwater, it's right at the interface. Hanalei Pier is just a couple hundred yards down the way, but it has fresh water from the Hanalei River coming in. Caffeine may be coming from all the people up the river. The pavilion has very little input from the river. Waioli Beach Park is further on down. Again, you have the restroom facilities there. Hanalei River itself doesn't have any measurable concentrations. So what you have at the pier is not coming from the river right there. It's coming from those huge restroom facilities that I just told you they were broken, and they put them on top of the land instead of burying them. Hanama'ulu Beach Park, big restroom facilities right on the beach. Kalihivai Beach does not have any restroom facilities at all on it. Kaloa Landing is a stream coming down into that area, and there's a lot of cesspools along the bank of the stream. It had really high, it was one of those that was really, really high in that first thing I showed you. Lumahai is a beach way on the North Shore. No restroom facilities, people just pee in the ocean out there. Nawili Willie Stream, I think this is great. That's another one of them that has really high bacteria counts, and I want them to put warning signs up. Look at this. Everything showing up in Nawili Willie Stream. Okay, this is important. Nawili Willie Stream empties into Kalapaki Bay. Okay, Nawili Willie Stream. Kaloa Landing. Okay, Poipu. Here, Poipu Beach Park on the South Shore. Remember Poipu Beach Park on the South Shore? That's probably it's a really small beach. It's like one of the only beaches on the South Shore. And it's where thousands of tourists go, and monk seals, which screw up your bacteria counts. A lot of monk seals, they wouldn't screw up caffeine counts, are on that beach. Salt Pond Beach Park, remember this, is in Hanama'ulu.